Hello everyone. So we need to tackle question 4D. We will do this in two separate parts. I need to give you some background about blood types in our species first. And so that'll be a 4D.1, and then we will actually do a codominance problem based on blood typing, and that'll be 4D.2. So whenever you talk about blood typing, you have to talk about a gentleman named Carl Landsteiner. So this will be 4D.1. Now at some point, you need to get yourself a hold of Google and the image search, and you need to look up Carl Landsteiner. He has one of the best handlebar mustaches that I've ever seen. You got to check him out. So his work with blood typing relates back to what we did with the plasma membrane. So if you recall, the plasma membrane is a phospholipid bilayer. So here's your two layers of phospholipids. And then there are integral membrane proteins that are embedded in the phospholipid bilayer. And then attached to these proteins are different sugars. And so I'm just going to draw some hexagonal sugars here. They could be glucose, they could be not glucose, fine. So here's your protein and here's your sugar. Well, whenever you attach a sugar to anything, that makes the compound a glyco. So if I connect sugar to this pen, it's a glycopen. So since you've connected sugar to a protein, this is a glycoprotein. And when it comes down to blood typing, it's all based on these sugar and protein complexes that are embedded in the red blood cell plasma membrane. It's all about glycoproteins. So Landsteiner studied blood, and he knew that if you injected blood from one person into another person, sometimes it was successful, and other times the blood clotted and the recipient died. And so he wanted to find out why. So if you take your blood and you put it under a microscope, there's going to be two kinds of cells. And when you put those cells under the microscope, if the light goes right through them, they are clear, and so they appear as white. And so we call them white blood cells. And then there's another group of blood cells that when you put them underneath the microscope, they're red. So we called them, ready, high-tech name, red blood cells. So there's red and white blood cells in your blood. The red blood cells are red and the white blood cells are clear. So the white blood cells are associated with your immune system. So your blood type is actually based on your red blood cells. These are nothing more than bags of hemoglobin, and their role is to take oxygen from your lungs to the tissue, drop the oxygen off for cellular respiration, and then pick up the waste carbon dioxide, actually the byproduct, and then take that CO2 back to the lungs and you breathe it out, and it's cellular respiration. Fine. So he discovered that there is a glycoprotein embedded in the surface of the red blood cell. Now, since this was the first glycoprotein he discovered, he called it A. Pretty high tech, huh? And then anyone who expresses this glycoprotein, he said that they have type A blood. And he kept looking. And hey, guess what? He found a second glycoprotein. He gave it a high-tech name, B. Thus, anyone who expresses the B glycoprotein has type B blood. He kept looking. And he found that some people express A and B glycoproteins in the plasma membrane of their red blood cell. So they have type AB blood. Then there were certain individuals that didn't express the A glycoprotein and they didn't express the B glycoprotein. So they didn't express either of these two glycoproteins, and so he called them type O. It's actually 0, but people call it type O. So one of the most popular ways to classify blood, and there are others, but the most popular way to classify blood is the classic type A, type B, type AB, and type O blood types. And it's based on the glycoproteins that are located in the plasma membrane of your red blood cells. Fine. Then, speaking of your immune system, There are Y-shaped proteins called antibodies, not antibiotics. Antibiotics are chemicals that kill bacteria. That's different. These are proteins, and these proteins are part of your immune system, and the job of this protein is to bond to cells or molecules that aren't from your body. And then your immune system literally attacks those foreign cells. So, anyone who has type A blood, what they will do is they will produce antibodies to B, because this is not you, right? If you're type A, B is a foreign protein. Therefore, we say that you produce anti-B. Well, let's say that you're a type B blood. That means that you produce antibodies to A. Thus, we say that you produce anti-A. Well, if you have type AB blood, you can't produce anti-A, or the antibodies will make your own blood clot. You can't produce anti-B, or you will clot your own blood, and you will have what we call an autoimmune disorder. So while you produce hundreds of thousands of other antibodies, you don't produce anti-A or anti-B. Okay, type O. It's the most common blood type. These individuals produce antibodies to both A and B. So we say that they produce anti-A and anti-B. So these are the antibodies that each blood type produces. So type A blood produces anti-B, type B blood produces anti-A, type O blood produces anti-A and B, and type AB doesn't produce antibodies to either glycoprotein. So why is this important? This is important for blood transfusions, okay? So if you have a type A blood person and they need blood, you can inject them with their same blood type. You can inject them with type A blood. If you inject them with type B blood, the antibodies will bond to this glycoprotein. Their blood will clot. It'll cause an aneurysm, an embolism, cause a heart attack. It'll kill them. It's a terrible way to go. You can't inject them with AB because the anti-B will bond here also. But what you can inject them with is O, because the antibodies are searching for the B glycoprotein. This antibody would charge this cell, searching for the B glycoprotein, doesn't see it, therefore there's no bonding. All right, someone who has type B blood, you can of course inject them with the same type blood. What else could you inject them with? You can inject them with O. Same ideas over here. These individuals produce anti-A. The antibody searches for the A glycoprotein, doesn't see it, no clotting. These individuals, you can inject them. Oh, I forgot the word inject. I suppose that would be helpful. Inject and inject. Now here's what's cool about being type AB blood. You can inject them with A, you can inject them with B, you can inject them with AB, and you can inject them with O. Ignoring the positive negative blood types, and we'll talk about this in just one second, you can inject any kind of blood, ignoring positive, negative, into these individuals. This is why they are referred to as the universal recipient. Universal recipient. They can receive any kind of blood. And there's a little bit more to that explanation. Whereas if you're type O blood, the only thing that you can inject into these individuals is O. That's it. Because I happen to be type O. If you put A blood into me, my antibodies will clot it and I'll die. You inject me with B, same thing. You inject me with AB and I'll die twice as fast. Okay, the only thing that you can inject someone with O blood with is O. Therefore, these individuals are referred to as the universal donor. And again, there's a little bit more to this story, and we'll come back and add more to this handout in just one second. Okay, So we need to then talk about, speaking of the positive and negative, we need to talk about what that means. So this is type A, type B, type AB, and type O. But maybe you've heard of like, oh, my blood type is O positive. I'm AB negative. Well, what does that mean? Well, it comes from a monkey. So what happens is we actually share a protein with another primate called the rhesus monkey. It's a little black old world monkey. Same thing, you should Google it, look up that little primate. Beautiful animals. So we match the protein with the rhesus monkey. And since this protein came from the rhesus monkey, or was first discovered here, we refer to it as the Rh factor. Rh from rhesus. Fine. So again, looking at the red blood cell. So here's the red blood cell that floats around in our blood. If it turns out that you express the Rh protein, we say that you are positive. This would be A positive, B positive, AB positive, O positive. Fine. Well, if you don't express the Rh protein, we refer to you as negative. But if you're negative, that means that you produce an antibody. That means that you produce anti-Rh. Okay? So let's take a look at what these red blood cells really mean. So let's draw 3 of these out. Let's take a red blood cell, and let's say that the person expresses, I'll just draw 4 of them, of course there's hundreds of thousands of them. Let's say that they express B and Rh. So put Rh and B like such. So what would this person's blood type be? Okay, their blood type would be B positive. Get it? B positive? All right, so if they're B positive, then that means that they produce anti-A. They can't produce anti-B, they'd clot their own blood, and they can't produce anti-Rh because they would clot their own blood also. All right, let's talk about my mama. Good old Grandma Kay. My mother's first name is Kay. That's where Emily's middle name comes from. So my mom's blood type is A negative. Okay? So if my mother is A negative, that means that she produces the A glycoprotein. But since she's negative, that means that she doesn't produce the Rh. Okay? So Grandma K is A negative. What does that mean? That means that in her red blood cell, she produces the A glycoprotein, but no Rh. Okay? So that means that my mom produces anti-B and anti-Rh. All right, so let's take a look at her son, her second son. I can write my own name. I am O positive. So since I am O positive, O means I don't produce the A glycoprotein and I don't produce the B glycoprotein. But what does the positive mean? The positive means that I produce the Rh. Like such. So, I know that Kay is my biological mother, but neither of us can donate blood directly to the other one. So here's my mama. Here's Kay, A negative. Here's her son, Robert, O positive. Okay? So, my mom goes in the hospital. Let's say that my mother needs blood. Can I donate my blood safely to my own biological mother? The answer is no. because my mother produces anti-RH. Don't forget she's negative, so since she's negative, she produces anti-RH. I express the RH gene, so I cannot donate to my own biological mother. What about the other way around? Can she donate to me? No, because I produce anti-A. Both of these are a bad idea. So I would donate to the blood bank, but it would be, I would donate 1 unit and then they would replace it and give my mother a safe unit. So let's go back to the statement over here about the universal donor and the universal recipient. How that really works, the universal recipient is really AB positive, because that means they don't produce anti-A, they don't produce anti-B, and they don't produce anti-RH. That is truly the universal recipient. Then the universal donor is actually O negative. Why? Because there's no A glycoprotein, there's no B glycoprotein, and there's no RH factor. So that's 4D.1, and then we'll take this information and tackle a codominant blood typing question.